Okay, so I started introducing Fisher's geometric model yesterday in a kind of qualitative way, and I want to do that more systematically now. So, so you'll see that the pictures that I'm going to draw will be rather similar to those that Maria just drew in the previous lecture. So we'll see about that. So Fisher's geometric model. And so as I mentioned yesterday, the model was actually not originally introduced to describe epistasis. As we discussed, Fisher wasn't really convinced of the importance of epistasis, but in recent years it has been used a lot in that context. So let me first introduce the elements of the model that are sort of classical. So we're considering a certain number of phenotypic traits. Right, and so the number of traits is n, and in Fisher's reasoning, it's important that n is large. As we'll see a little bit later, in some sense, it doesn't really matter that much whether n is large or small. But and these are real valued phenotypic traits. And they form a vector, right? So we have x1 up to xn, which I will denote by a vector. And so this is an element of the Euclidean n-dimensional space. Fitness is a function of these traits, and it's a simple function. So it's a simple function with an optimum at the origin. So fitness is a function of f of x with a single optimum at x equal to 0. And it's not really important for what I'm going to talk about, what this function looks like. But for example, something that is often being used is a Gaussian function. So you can write this f as e to the minus x squared, maybe with some scale that I will call 2 lambda, but that doesn't really, it's not going to, or lambda squared maybe. But this doesn't really make much, so this will actually not be very important, as you'll see. And so this is the fitness, and now mutations occur, and mutations, the assumption is that the mutations are random displacements in the phenotype. And in Fisher's original argument, he assumed that these displacements are isotropic. So this is something that is often called the assumption of universal pleiotropy, which means that every mutation affects to some degree all the traits, right? So mutations are isotropic random displacements, random changes of the phenotype. Now, in Fisher's original argument, he actually assumed that these displacements all have the same size. So there's a certain scale of mutational displacements. So I'm going to assume that the displacements are actually Gaussian random vectors. So there's some probability distribution. So M is a mutation vector, and there's a probability distribution, which is an isotropic Gaussian. And so the probability of having a certain displacement is e to the minus m squared divided by sigma squared, where sigma squared is the variance of the mutation. So that sort of tells you how big the mutations are. Okay. Now, so this is sort of apart from the small change with regard to how we model the mutations, this is essentially Fisher's model. He introduced it in his famous book in 1930. And the motivation or the question that he wanted to ask, so Fisher's question was does evolution proceed in large or small steps? And more precisely, the question that he was asking was, given this scenario, and given that you have a certain amount of maladaptation, so again, I'm going to draw my pictures for n equal to 2. So this is our phenotypic space. We have lines of constant fitness. I could make these, elliptic, like in Maria's talk. So you could, in principle, here introduce some anisotropy in the fitness function. You could also introduce some anisotropy in the mutations. To some extent, these can be scaled away. But here I'm just going to assume for simplicity that everything is isotropic. And so Fisher's picture was the following. So suppose there's some degree of maladaptation. So my organism is initially sitting here, right? So this is my initial phenotype. And now I look at the mutations, and these mutations, as I said, are random displacements, and they will cover, so my random mutations will be covering some circle, and as I said, Fisher assumed that they all have the same size, so then this is a circle of diameter 2r. And the question is, what is the probability that one of these mutations is beneficial, okay? As a function of this degree of maladaptation, and so let's say this is the distance of my phenotype from the optimum initially. So this distance here, let me call it d. So what is the probability that a mutation is beneficial? Given the degree of maladaptation, d, and the mutation scale r. And so this you can calculate. Actually, the calculation is not entirely trivial, and it's one of the, I think, one of only 2 or 3 equations that appear in Fisher's book, and he just writes it down. And I think the derivation appeared like 20 or 30 years later in some textbook, and even today there are research papers appearing saying a new derivation of that equation. So it's not, it's not, but this was sort of his style. So this probability, let me call it PB, so there's a probability that a mutation is beneficial. And so Fisher's calculation is this. So this is technically, it's an integral over a Gaussian. And so there's a parameter small, that I call small p here. It's an integral over a Gaussian. Technically, this is something like the complementary error function. So this is his answer. And this p is a combination of the parameters. Specifically, it's the mutation scale times the square root of the number of traits divided by 2d, okay? And so the key point is that, and this was sort of Fisher's key conclusion, so you might think that this only depends on R over D, right? Obviously, what you can sort of see from this picture, if you go further away, then you will have more and more beneficial mutations. If you're very close, then of course it's very likely that you will overshoot the optimum. But what is sort of non-trivial about this calculation is this additional factor of square root of n. And this is, I'm drawing the picture for n equal to 2, but of course things are sort of, change with higher dimensions. And so the key point here is that even if you keep R over D fixed, so suppose you say, okay, if I'm at a distance d and I keep my mutational scale at 10% of that, I should have a good chance of finding a beneficial mutation. But this is not true, because as n gets big, this goes to 0, right? So then the lower boundary of the integral moves to infinity, and this goes to 0, right? So you need, when n is large, you need much, much smaller steps to find something that is beneficial than you would expect. I mean, that's really the key point. So, so to have a reasonable fraction of beneficial mutations, which essentially means that you want to you want P to be some constant of order 1. You need that this P is of order 1, and so you need that the mutational displacement should be of the order of D divided by square root of N. And this is much, much smaller than D when N is large. Okay, so this means that if a complex organism, for a complex organism to be able to evolve, it can only do this in very small steps. And Fisher, gives some hand waving or some qualitative arguments for this. It's like a microscope, and if you have to, you have many knobs that you have to tune, and so if you change one trait, you're likely to make another worse. And so this subtle problem leads to this behavior. Okay. No, sigma squared is just related. I mean, so I'm calling this R. So essentially, R is equal to square root of N times sigma, right? So that's just the mutational scale. I mean, how big the circle is, right? So this is just. I mean, you can replace it by. I mean, this is sort of, a matter of taste whether you want to consider the size of the overall mutational displacement as your basic variable, or the size of the displacement in one component. They differ by a factor of square root of N, but in either case, so if you replaced R by sigma, then you would have a 1 over N here instead. But the qualitative argument would be the same. Right? I mean, here I'm just, here I'm assuming these displacements are random. So typically, the area that you can cover by a mutation is sigma times square root of N. Yeah, but it only depends on the scale of the distribution, right? So the scale of the distribution is exactly R, right? It's a mutational scale. But the point is, I mean, what is sort of hard to visualize, the point is that in high dimension, I mean, this is, the mechanism behind this is that if you look at random displacements in high dimensions, so, you have many dimensions, you have random displacements, you have one dimension that leads towards the optimum. So in high dimensions, essentially all of the random vectors will be pointing perpendicular to this direction. So the vectors will be, if you pick a random vector, most likely it will be pointing in this direction. And so this will always give you a deleterious mutation because of the curvature of the fitness isoclines, right? So it will not. So it becomes more and more difficult to generate something that actually points in the direction where you want to go. Yep. Of course. Yeah, that's right. Yes. Yeah, well, you'll see the Brownian motion appear in very shortly. I mean, so the Brownian motion, Brownian motion means, and that's what we're going to do next, is that sort of add many of these displacements. And then, the issue is, can you actually find this place, the center? And that, of course, also becomes harder and harder as N gets bigger. Yeah. Okay. So this is sort of Fisher's classic argument. So his conclusion was that mutational steps have to be extremely small. Now this was before people fully understood the effect of genetic drift. So from Daniel's lectures that there's a fixation probability which is proportional to the selection coefficient. So very small mutations of very small effect tend to be lost by genetic drift. So in fact there is a kind of trade-off, and that was pointed out later by Kimura, that if you take into account that these very small mutations, they may be beneficial, but they will be lost then you actually find that the most relevant mutations for selection are sort of intermediate effect, right? So they shouldn't be too large because of this, but they also shouldn't be too small because of genetic drift. Yeah. Okay, I mean here, this is of course not, this is not really a distribution of fitness effects. I mean, you can calculate a distribution of fitness effects from this model, and then what you actually find, and okay, this is sort of fairly trivial. I mean, if you look at the beneficial side, then because the way the fitness is defined here, there's of course a cap, right? I mean, there's a maximum fitness, but and this is actually something that Orr showed some time ago, so this was sort of part of the revival of the Fisher's geometric model. So Orr showed that if you're far away from this maximum fitness, then typically Fisher's geometric model will give you an exponential distribution of fitness effects, and this is, this was considered at least for a while after these papers by Orr to be the typical situation. I mean, now we know that there are of course also more heavy-tailed distributions, which are probably more difficult to accommodate in this model, although you could have some funny shape of the peak and then you could even accommodate that. Okay. Right. Yeah. Well, I mean, so just, so this is the effect size, but of course it's the effect size of these phenotypes that you probably don't even know what they are. So the fitness distribution, the distribution of fitness effect size is different, and there are a number of papers that have compared the fitness distribution that comes out of this with experiments, and in some cases it seems to work quite well. Okay. Right. And of course, there's a big issue here what this n is, right? I mean, this is, and we'll get to that because I'll show you some data where of course, once you have this model, you can try to estimate n, and then, what does it mean? So I think, I mean, you're from Montpellier, right? So Montpellier is like the hub of activity on Fisher's geometric model these days with Guillaume Martin and other people there. And so I think he has very nicely shown that the way to think about this is that this n is really the trait, as you say, the traits that contribute to selection. I mean, it's probably not all the traits of the organism, but it should be thought about as those that are sort of relevant for fitness. Okay. Okay, so this is sort of the classic story, but as I said, the recent application of this model is with regard to epistasis, and for that we have to add a fourth assumption, which, as far as I know, appeared first in this context in a paper by Guillaume Martin and collaborators, which was precisely concerned with actually the distribution of epistatic effects in comparison with data. And this assumption is that phenotypic effects are additive. Okay, so this is additivity of phenotypic effects. And so the assumption here is that if I now combine mutations, so the combined effect of two mutations let me call them M1 and M2, is just the vectorial sum of the two. Which means that you add all the components separately, and that gives rise to the kind of picture that I showed you yesterday, where you have these arrows in this plane, and you can see how epistasis and sign epistasis proceeds. And so to sort of connect this to what we've done in previous lectures, with this additional postulate, we can now define a genotype-phenotype-fitness map, which is what Kavita asked about yesterday. So with this, we can sort of directly map the L-dimensional genotype, which is a sequence of zeros and ones. Right, so this is a sequence of zeros and ones indicating whether a certain mutation is present or not to fitness in the following way. So we want to have this fitness function. And so we just have to we have to apply our phenotypic fitness function to the phenotype. And what is this phenotype? Well, we'll start, let me draw another picture here. So we start at some initial phenotypic point, which I will call Q. So this is my wild type phenotype, and then I do a mutation. Okay, so this is my first mutation, then I add a second one, and a third one, and so on, right? So these mutations now form a kind of path in this phenotypic space, and which mutations are present is determined by the tau. So the argument of this function is the Q plus a sum from 1 to L times tau I times mi, where these mi's now are a fixed set of vectors, right? So these mi's are a fixed but random, so they are drawn from this distribution, random set of mutational displacements. For I from 1 to L, so I have to pick L mutations, and then how many of those I include in the sum is determined by the tau sequence. Okay, so this is now my fundamental mapping. And so this will generate for me a fitness landscape on the space of genotypes for every set of random vectors that I choose. And these random vectors are drawn from this Gaussian ensemble, so I can now calculate average properties of these fitness landscapes in this way. Okay. Is that clear? Okay. And so this is the background. So let me just give you a very brief summary of the results of this. Yeah. But here, I mean, throughout this week there has been, in my lectures, there has been no environment, and so the assumption is always, in some sense, we fix some environment, and this is what we study, and we assume that it doesn't change, right? Sure. Yeah. And this is sure. Yeah. I mean, and this has of course been studied quite a lot. So you can you sort of have a moving optimum, and then there's some issue of whether adaptation is able to keep up with this, and so on. So these things, of course, can be done. Yeah. Right. Okay, but so here, what I want to focus on is, what does this tell us about epistasis and complexity of fitness landscapes? So the key results here, and so this is, essentially the content of this paper that is also in the reading material in genetics. And so what we were primarily interested in here is, again, just as a quantifier of the complexity of this fitness landscape, is a number of local maxima in this fitness landscape. And so what does this depend on? So the number of fitness peaks. And it depends essentially on two parameters, or three. Of course, it depends on L, as usual, the number of loci. It depends on the phenotypic dimension N, and it depends on the degree of maladaptation, which is just the distance of this initial point from the optimum. And qualitatively, what is happening, and I'll make this more precise in the moment, is that the closer you are to the peak, the more rugged the landscape is, right? If you're very far away from the peak, then epistasis is very weak, essentially because then the curvature of these fitness isoclines is low. And so essentially everything, far away from the peak, these isoclines essentially look like straight lines, and then everything is linear, okay? So this depends on Q, but of course, the only thing that matters is Q divided by sigma, so relative to the mutational displacement, right? So essentially, what you need to know is how many steps would you have to take to get to the optimum if you could use the shortest path. And what do we find? So the expected value of the number of maxima, it again grows exponentially, but there's a power law prefactor. And then there's an exponential growth rate, which we call the complexity. And the small q is simply the big Q divided by sigma and L. So it turns out that you have to scale, which is also not so surprising. If you have L mutations, then of course the distance that you can cover is more or less sigma times L. So you have to scale by that, and then you make L very large, and this function sigma of Q is not, it's given by a variational principle that I don't want to write down, but it has a relatively simple shape. So this is Q. So it starts out as at log 2. So log 2 is the largest value that it can have, because then this is 2 to the L. So this means that essentially a finite fraction of the sequences are maxima, and then it decreases, and it goes to 0 at some critical value, QC, which is approximately 0.92. Okay, so as you go further away from the optimum, the landscape becomes less and less rugged, and eventually, here there's another phase where things are still sort of interesting, but I don't really have time to go into that here. But the main point is that in this region here, the number of maxima is no longer exponentially large in L. Now, one interesting and to us surprising result here is that the dependence on N, that is on the number of phenotypic traits, is relatively weak. So this function sigma doesn't actually depend on N. The only N dependence is here in the prefactor, which is rather weak. And this is, coming back to the remark before, this is really related to Brownian motion. So this is essentially, this N over 2 comes from the probability of a random walk to find the origin. This you can sort of see from the derivation. But apart from that, there is a little dependence on N. And moreover, if you ask in what direction does the dependence on N go, you see that if you increase N, actually the number of maxima goes down. Okay, so more phenotypic traits does not mean that the landscape is more rugged. Okay, and this dependence is decreasing. That is larger N implies somewhat less rugged landscapes. But the most important determinant of the ruggedness is the distance to the optimum, so the Q. Another thing that I wanted to mention, because it came up, I think, a few days ago. So there was this question in mathematical terms, to what extent this quantity, the number of peaks, is self-averaging. That is, to what extent the mean is informative of the typical behavior. And it turns out that in this problem, there is at least a region where these two quantities are rather different. So the typical value actually behaves differently. So if I plot, so the sigma is essentially the log of the number of maxima divided by L, right? This is more or less what it is, of the average number of maxima. Now, if I instead calculate the log of the typical number of maxima divided by L, it looks something like this. And it goes to 0 at a different value, which is 1 over 2 times square root of pi, which is smaller. And so you have in between, you have a region. So here you have a region where typical landscapes don't have a lot of maxima, but a few landscapes have very many, right? So here you have a kind of coexistence of two different landscape structures. And we think that this has some relevance because people have tried or are trying these days to use Fish's geometric model to fit to data. And then, of course, if you're in a parameter regime where the same parameters can describe landscapes that will look very different, such an inference will be very hard. And this seems also to have been observed in some of these studies. So let me show you a couple of slides. So can we start the projector? Yeah, so I'm going to show you an example. I mean, given this model, of course, you can try to infer it. So I'm going to show you an example where we get a number out. I'm not, I think it's difficult to interpret that, but in principle it can be done. So let's see. Right, so this is a picture that I showed you yesterday. This is basically illustrating this mapping, the mapping from the genotype to the phenotype and then to the fitness via these lines, right? So this is our Q, and this is what these little landscapes, which are projections of the hypercube, look like. And in the next slide, you see two instances. So this is now. This is a case, so there's L equal to 6, N equal to 2, and this is a case which is sort of in this coexistence region. And so you see that for certain realizations, you get something like this. So what is plotted here, this is again a rendering of the 6-dimensional hypercube, and what is plotted here is essentially fitness. So what is written here is the scale distance to the optimum. And so this is the wild type, and these red things are local maxima, right? So you see here on the left, first of all, this landscape reaches rather high values of fitness, so these phenotypes are close to the optimum, and it has many maxima. This one doesn't get that far and has much fewer maxima. So this is a kind of dichotomy between the landscapes that we see here, because, and this is sort of part of the derivation, I think you can, it's rather easy to understand and explain in the paper, that in order to have a landscape with many maxima, you need to get close to the optimum. Okay, if you're far away from the optimum, you won't get that. Okay, so this is essentially what these landscapes look like. Now I want to Just to illustrate one experimental application of this, not looking at full landscapes, but looking at pairwise epistasis, I want to briefly talk about a study with the experimentalists, Simon Schoustra and Arjan de Visser, which is also in the reading material. So this was a study of mutations in a fungus, Aspergillus nidulans. And so what Simon did was to collect 244 beneficial mutations, and essentially he did this phenotypically. So he grew colonies, many colonies in 2 different media, and he sort of picked out sectors looking like beneficial mutations, and you know, had various ways of verifying that they were different and so on. So in the end, he had 244 beneficial mutations, and the goal of this study was to see how the epistatic interactions between pairs of these mutations depends on their overall effect. So this is similar to what I talked about 2 days ago. So again, one can use the complicated reproduction schemes of this fungus to so use sexual crosses in order to put these mutations into combinations. So he picked pairs of mutations from his sample that had similar effect, and put them together, and then measured the fitness of the pair. And then he defined the pairwise epistatic interaction the way we typically do. And so this is a case where the fitness effects of the 2 individual strains are closely similar. So we can basically look at this quantity as a function of the single mutation strength. Now there is in the essay that Simon used, there is a kind of technical problem, which is that he couldn't really, so after having mixed the types, he had a colony, or he had a sample containing all 4 types, so he couldn't really measure the double mutant fitness by itself, and he could only detect it if the double mutant fitness was larger than the 2 single mutant fitnesses, which actually means that one cannot directly determine sign epistasis in this case. And because these 2 things are equal to S, sign epistasis here means that epsilon AB should be less than minus S. So those we don't actually see, and that had to be sort of factored into the analysis. And so you know, just this is sort of similar to what I showed you yesterday. So just to show you what Fisher's geometric model in principle tells us about this problem. So here we have 2 pairs of mutations which have the same effect size, right? So they go to the same circle at the same distance from the optimum, and one of them displays magnitude epistasis. I mean, this you basically always get simply because the fitness surface is curved. So it's clear that, you know, you will have less than additive fitness gains here, right? So this will give you negative magnitude epistasis. But here, you know, this is the same selection coefficient, but you overshoot and you get a negative effect. So here you get a reciprocal sign epistasis. And so the point here is that pairs of mutations that have the same fitness effect differ widely in their epistatic interaction. So this model does not predict, for a given single mutant selection coefficient, it doesn't predict a particular value of the epistatic interaction, but it actually predicts a distribution, okay? And so the model in that sense contains a mechanism of intrinsic variability, and that actually turns out to be important when you analyze the data, so you can sort of compute from the model a prediction for the distribution of epistatic effects conditioned on the single mutant effects, and you can fit it to the data, and this is what this looks like. So here we have the epistatic interaction as a function of the selection coefficient normalized by the largest selection coefficient that was observed. Here is 0, so you see that typically, you know, these interactions are predominantly negative. There is this line minus S here, so the measurements below here in some sense shouldn't be there, because I told you that the assay doesn't allow them to actually see growth rates that are, you know, instances where the growth rate of the double mutant is smaller than the growth rate of the single mutants. They are nevertheless there, and this is some complication which has to do with the fact that sometimes a very slowly growing variant can dominate by somehow germinating quicker than the others, right? So you germinate quicker, you sort of suppress the other mutants, and then you can still grow. But these are sort of exceptions. So you know, most of the ones that actually have sign epistasis tend to sort of crowd around this -S line. Okay, and so this was analyzed using a maximum likelihood approach based on Fisher's geometric model. And these two, the blue region corresponds to the variability from Fisher's geometric model. The pink region corresponds to what we estimate the measurement error to be. And what you can sort of see is that the measurement error doesn't really explain the variability, so you need this additional intrinsic variability provided by Fisher's geometric model. And doing that, you can estimate the parameters. And so essentially, you know, this is what they are. So there are actually three parameters. There is the distance from the optimum relative to the standard deviation of the mutations, which is about 7 or 10. This is the number of dimensions, and this is the sort of the ratio of the true limit of fitness. So S0 is essentially the selection coefficient that you would get if you went all the way to the optimum, and Sm is the maximum selection coefficient that is actually observed. So it's a kind of consistency check that this number is greater than 1, because if it were smaller than 1, then the model actually wouldn't really be consistent. But this is what you get. So, you know, so n is whatever, 20 or 35. I'm not sure that this really, you know, tells you much. But what we saw, of course, found interesting, or what was maybe surprising initially, was that you have a smaller n in rich medium than in poor medium. But this was the expectation of things to be the other way around, is of course in some sense based on the expectation that having more phenotypic traits means in some sense more complexity. On the other hand, as I explained to you before, in terms of the fitness landscape, the genotypic fitness landscape, there's actually less complexity when n is larger, which is sort of more consistent with this ordering. But again, I wouldn't put too much, you know, put too much meaning into these numbers. It's just showing that the model can be used to consistently describe data of this form. Okay. All right. Are there any more questions about Fisher's geometric model? Yes. Yeah, but it's sort of all over the map. I think there are even some studies that find n smaller than 1. Yeah, I'm not, you know, I don't have an overview of what has been, you know, what has been. I think probably in this review paper by Tenaillon, there's maybe some more information about that, but I don't have it top of my head. I cannot really tell you. I mean, I know that there are studies that find much smaller values of n, but I think there are also, I mean, Dan Weinreich did a study recently. Yeah, okay, but he probably also found very small values of n, I think. But I'm not, you know, I wouldn't be able to tell you what is. My feeling is that there are probably also cases where it's much bigger. But I mean, one thing that we noticed when doing this analysis is that, you know, just in terms of inferring things, in terms of the maximum likelihood function, this inference problem is not very well, it's not very stable. So what you typically find is that there are certain combinations of n and d that give almost the same quality, right? So there's sort of a manifold in the parameter space where the likelihood doesn't change much, and therefore pinning down these values is at least in this case was rather subtle. Okay. Okay, so if there are no more questions about this, then I would like to spend like the last half hour on the 5th topic that I had promised to you. So there will not be that much time, but I think I can get something across. which is about, so what I called this lecture was navigating fitness landscapes. So far, we sort of talked about the structure of fitness landscapes and epistasis and so on. Now, of course, the question is what is sort of the dynamical content? How do populations actually evolve on these fitness landscapes? And so let me spend a little bit time explaining just two sort of simple but paradigmatic kinds of models that people have used here. So if you could turn off the projector again, okay? Yeah, he's doing it. You don't have to go up. And in some sense, this task is not so difficult because Daniel already gave you a good introduction to the regimes of evolutionary dynamics. And so what we're going to consider is a regime of weak mutation, right? So this is the weak mutation regime. Which essentially, means in some sense that the population size times the mutation rate. So for me, U is the number, the mutation rate per individual and generation of any kind. So I want this to be very small. Okay, how small exactly would depend on other things, but let's just say we're in this weak mutation regime. And as Daniel explained to you, in this weak mutation regime, what happens is essentially that once in a while a mutation appears, and then it either goes to fixation or it goes extinct, and then for a long time nothing happens, and then another mutation appears. So we have basically a process which consists of the sequential fixation, or not, but those events we don't really care about, of mutations. And so if we think about this in terms of our fitness landscape picture, all right, so this is our space, and I'm now going to label these genotypes just by indices. So let's say this is a genotype I, so the population is currently sitting at this genotype I. It's creating some mutations, and perhaps one mutation fixes. And so let's say if it fixes, then it sort of goes to another genotype J, right? So this is sort of the elementary step, and only single mutations can fix. So this thing sort of is moving around on this hypercube in single mutational steps, right? And so mathematically speaking, this is a Markov chain. This is a Markov chain on the hypercube, or on the sequence space. And the rate at which this happens is of course proportional to the rate at which mutations appear. But that I will sort of factor out. So I will assume here that I will sort of measure times in units of the mutation time, which is 1 over n times u. So I will basically, I will just scale time so that mutations appear at rate 1. And then the rate for such a transition, from I to j, is just a fixation probability. And I will use, Daniel, emphasized that that one shouldn't pay too much attention to the precise form of this, and there are universal and non-universal features and so on. But just to be, to fix ideas, I want to use a particular formula. And this is the Kimura formula that I introduced already in the first lecture, which sort of contains all the regimes that we need. And this is what it looks like. All right, so here we have the fitness difference, and this is a selection coefficient and the population size. So this is a Kimura formula. Okay, so this specifies the model, right? So if I give you these fitness values, then you have this Markov chain, and it's sort of moving around on this hypercube, and this is it. Okay. And I should just emphasize that this formula is valid for all the cases. So either if fi is greater than fj, fi is less than fj, and even for the neutral case, you'll check it just gives you 1 over n, which is also the correct result. Okay, so now one can look at two different kinds of dynamics. So what you will notice here is that this formula, of course, always applies, this is non-zero, whatever these fi's and fj's are. So this always describes mutations, beneficial mutations as well as deleterious mutations. So it's actually a dynamics that is reversible. And in fact, in terms of Markov chain theory, this is a reversible Markov chain, and this is what I want to show to you. So how do you, generally speaking, how do you, what do you do with a Markov chain like this? So what you would like to know is probably what is the probability. So let me call pi of t. Let me call this the probability that the genotype at time t is equal to I. Okay, the probability that the the population in this picture is always monomorphic, so it's sort of jumping around. So the population is at I at time t. Now this satisfies something that is called a master equation. So who has never heard the term master equation? A few. Okay, so I'm going to write it down. So it's actually not very deep. I mean, what you have to do, you just want to keep track of how the probability evolves. So you just have to figure out, to see how this probability evolves, you have to figure out what are the possible processes that have that allow the population to enter this or to exit from it. So it's a kind of balance equation. And so what you just have to write down is dpi by dt. And here you have to sum over all the genotypes that are at distance 1. Okay, so this is dij equal to 1, where dij is the Hamming distance between the two genotypes. And then we have a kind of inflow of probability that is stuff can go from j to I. And this happens with a probability that is given with the probability that it is in at j before. And there is an outflow term, which is just pi j times pi. So this is the master equation, and this governs how the probability changes. And the most fundamental thing that you might want to know is what is a stationary distribution. So suppose, this process of sequential mutations is going on for a long time. So the population sort of samples this little space, and it will, eventually reach some sort of a steady state. So the question is, what kind of distribution do I need to make this vanish? So this is for in the so-called stationary state. Okay, so what distribution pi star, which is my stationary distribution, can I find that makes this vanish? Okay, and you can sort of see it by looking at this. You can see that a simple way of making this vanish is if each term vanishes. Okay, if each term in the sum vanishes, this is obviously a sufficient condition for the whole thing to vanish. So what we can do is that we look for a stationary distribution that satisfies what is called a detailed balance condition. Or in the mathematical theory of Markov chains, this is called a reversibility condition. And this detailed balance condition simply means that each term in the sum should be equal to 0. And so what I want is that Pji of Pj star is equal to Pij times Pi star. Which means that the probability fluxes between any two genotypes are balanced, right? So I have as much processes going one way as the other. Okay, now this we can actually easily solve. So let's just put the pi's on one side and the pi i's on the other. And then I just insert my Kimura formula. And I'm going to call the selection coefficient in this just to make writing a little bit easier. I'm going to write S is equal to Fi minus Fj. So this is my current selection coefficient. So then, or maybe, okay, in my notes I have Fj minus Fi. So let me do it that way. So otherwise I might make a mistake. Okay, so what does this give us? So the first Kimura formula gives us e to the 1 minus e to the 2s, because s is now the selection coefficient from j to I and not from I to j. And downstairs I have e to the minus 2ns. Okay, and then I have to have 1 over the other Kimura formula, which has a minus 2s. But here it has the n. And here it has no n. Okay. No, well, I'm just considering one transition, right? So I'm considering one mutation and the back mutation, so there's only one s. Okay, and now you can sort of manipulate this a little bit. Obviously, you want these things to cancel, so you can pull out the e to the 2s here. So then we have e to the minus 2s minus 1. And here I pull out the e to the minus 2ns, and then I get e to the 2ns minus 1. And downstairs I don't do anything. And now you see that the factors upstairs and downstairs cancel. There's a sign change, but because there are two sign changes, those cancel as well. right? So I can cancel this against that, and I can cancel this against that. And so what I get is e to the minus 2n minus 1 times s, but s is fj minus fi. Okay. And so you see that the ratio of the probabilities is just the exponential in the fitness differences. And so I can just, attribute each fitness to one of these probabilities. So what this tells me is that the stationary fitness pi star of a genotype is proportional up to a normalization factor 2n minus 1 times its fitness. So this is a very useful formula. And of course, in some sense, it tells us that, that the model makes sense. What it tells us is that selection, of course, is supposed to concentrate the population at a high fitness genotype. So this tells us that the occupancy at the genotype is sort of exponentially enhanced by a factor that contains a population size under fitness. Okay. This is a very useful formula. I think it was, as far as I know, it was first pointed out by my colleagues Johannes Berger and Michael Lessig in Cologne in 2004, but then also sort of rediscovered and put into a more general framework by Seller and Hirsch in a paper in 2005. Of course, a statistical physicist will see that this is actually nothing but a Boltzmann distribution where the analog of energy is minus fitness, and the population size plays a role of inverse temperature. Okay, but this is just, for as a side remark. But this is useful because it actually, what it means is, at least if you're in this situation, so if you're in a situation where, you sort of have reasons to believe that selection has acted in a constant way over a long time, and this, the space that you're interested in actually has been explored, then you can basically, by just looking at how frequent different genotypes are, you can infer at least the product of population size and fitness, right? So this allows you to sort of infer n times fi from abundances, so to speak. And this is actually being used in various contexts nowadays in quite successfully. Okay. Yep. Yeah, it's sort of and so on. But I certainly think that there are contexts where it applies. Of course, there are contexts where it doesn't apply, and then you have these complications of clonal interference and so on that Daniel talked about. Yeah. Yes. Well, no. Within a population, there shouldn't really be. Well, no, but or over time, right? Of course, you can sort of, if this picture is right, then the population will sort of change over time, and it will sort of wander around in this landscape. Well, I know. This. As I said, Berger and Lessig, they developed this in the context of studying transcription factors, and sort of on the molecular level, I think you can have a lot of data like that. So if you but I don't, I don't know whether it makes any sense for natural populations in the wild or anything like that. I don't know. Okay, so this is one regime. Now, this, of course, so as I said, this is sort of the reversible case, right? So here, the assumption is that on the time scale of interest, both the forward and the backward mutations can occur. And when n is very large, this is of course not really true. So there's a second regime that has also been studied extensively in theoretical population genetics, where one assumes that selection is so strong that that these reverse transitions don't happen. I mean, you will notice that for this dynamics that I just described, these accessible pathways that I spent a lot of time discussing previously don't really play any role. I mean, here the thing is sort of exploring everything, the population is exploring the whole space, and it doesn't really care if fitness goes up or down in the long run. Although, of course, it tends to be, concentrated in regions of high fitness. Now, the second regime that I want to describe is closely related to these accessible pathways. And these dynamics are called adaptive walks. Or this is also often abbreviated SSWM, which means strong selection, weak mutation. So here we sort of put another assumption in. So we assume strong selection, weak mutation. We assume that typical selection coefficients are such that the product of the selection coefficient and the population size is very large, right? Which basically means that this factor down here either can be neglected if the mutation is beneficial, or it's very large so that the whole fixation probability is zero if the mutation is deleterious. So this means that I basically put all the Pij is equal to 1 minus e to the minus 2 fi minus fj if fi is greater than fj. So for beneficial mutations, the fixation probability becomes independent of n. So actually the population size drops out now. So this is independent of n. And it's simply zero if the mutation is deleterious. Right? And then, of course, the dynamics changes. So then the population can only move along these accessible pathways. So the population performs so-called adaptive walks, or an adaptive walk. Along these accessible pathways. And one thing that this allows you to do is to put weights on these pathways, right? I mean, so in previous lectures, we sort of discussed whether these pathways exist and how many there are and so on. But we haven't really been able to say which pathways are sort of really important. And with this, we can do that. So weights of pathways. And so what we need to know, so suppose we have, the last cube I will draw in this lecture, maybe. So suppose we have some pathway on this graph that does something, right? So it's accessible by definition. And so at each juncture of this path, like in the first point and the second point and so on, there will be a certain number of directions in which it can go. Maybe there's only one, then it will, it has to go there, but maybe there's more than one. So you have to calculate the probability, which I will call T. So T from I of j is the probability that the transition from I and j will occur, conditioned on there being such a transition. So we don't care about, the time that the population is sitting here and waiting for a mutation. We just want to know what is the probability of such an adaptive walk. Adaptive walk goes from I to j, given that it goes at all, right? Given that a step occurs. And what Gillespie showed, and this is actually very plausible, and it's easy to sort of prove formally, what you have to do is you have to take the fixation probability of that particular step, and you have to normalize it by the fixation probabilities of all the other steps that could have occurred. So you have to sum here over your neighboring genotypes, PIK, where the sum runs over the genotypes that are at distance 1 and that have higher fitness, right? So this could be 1 if there's only one possibility, or it could be very small if there are many other possibilities, and so on. So this is the Gillespie. And so with this now, so now you basically have, for each such path, you know the probability that that particular step is taken. And so now you can just define a path weight by taking the product over these transition probabilities. So the path weight so suppose I have a path going from I1 to I2 and so on up to IL. The path weight is just, so this is now the path from I1 to I2 and so on up to IL. It's just a product of these things, right? T of I k to I k plus 1. And if you remember in some of the I think the landscape, the famous Weinreich landscape that I showed you, there were sort of arrows of different thickness, so there was a kind of highlighting of which paths were more important, and this is exactly by this method. So this way you can put a number on the pathways, and in many of the empirical fitness landscapes, one finds that in fact the probability is sort of concentrated on a few pathways. So not only are there few accessible ones, but also among the accessible ones, some are much more likely to be taken than others. Now, of course, these paths, by definition, they will always terminate at some point in a local fitness maximum, right? Once you have reached the local fitness maximum, you cannot go anywhere. And so, as a way of quantifying how rugged a landscape is, you can just ask how far do I get, right? I can perform an adaptive walk, and I can ask how far can I actually get. And for that, I would like to show another slide. So if you could please turn on the projector once more. So as I said, okay, very good. So as I said, adaptive walks. Walks terminate at fitness peaks. So we can use the length of an adaptive walk, the number of steps, to quantify ruggedness. So this, of course, I mean, with regard to the models that I have been introducing, this poses a question: how long are adaptive walks in typical model landscapes? This is a topic that Kavita has done nice work on over the years. So basically, what you find from the models is that the typical length, so let me call this L, right? So the typical length of an adaptive walk in the House of Cards model, the length is typically log L, proportional to log L. So they are very short, right? You know, the maximum distance you could go is L, but you only go log L. In the NK model, they are typically something like L divided by K. Some corrections. But I wanted to show you, you know, coming back once more to this experimental paper by Claudia Banca and collaborators, I want to show you how you can sort of make use of this for the analysis of data. And this is quite, you know, quite useful in particular for data sets. I mean, this data set is not that large. It has 640 genotypes. So they were actually able to determine all the maxima, but there are also data sets that are much larger where you cannot even computationally enumerate all the maxima. And so then what you can do is to basically just send out an adaptive walk and see what it finds, right? And so this is now again from the Claudia Bank paper. And the first plot shows you what they call the absorbing time. So they actually also in this paper develop a very nice algorithm to efficiently basically compute. So they don't actually simulate these adaptive walks, but they efficiently compute the adaptive walks properties directly from the data. These are the expected absorbing time. So this means just what I call the length of the walk, right? So you basically start adaptive walks from all possible places, genotypes, and you let them run and you ask how many steps do they take. And so here the largest absorption time is 8. And this red line is what the absorption time if you start at the parental sequence, also at the wild type, if you will. And then they asked, what is a probability that starting at a certain genotype you will reach the global maximum, right? So the probability of being absorbed at the global maximum. This is again across all starting points. And so you see there are quite a few, there's a fairly large fraction of starting points from which you can reach the global maximum, which shows you that it's not, the landscape is not that rugged. But if you start from the parental sequence, then you only have about less than a 40% or 30% probability of reaching the global maximum. And this is because there are other peaks that are closer. So this is another peak. So this is the global maximum. So the red ones are substitutions relative to the parental sequence. So this is at distance 4. And there's another lower local maximum at distance 2. And this is, generally less likely to be reached, but it's more likely to be reached from the wild type parental sequence. Okay, and so as a last word, so if there's of course much more, or would be much more to say about these adaptive walks. If you're interested in this model, so the last problem on the problem sheet is essentially about calculating this thing. So you want to show that the length of the adaptive walk is of order log L. So this is problem 7, I think. But this actually treats a slightly simpler variant of this model, which is called the random adaptive walk. And the random adaptive walk is the approximation or the limit of this thing. So if you imagine that all the beneficial selection coefficients would be very large, so then these fixation probabilities, provided the mutation is beneficial, the fixation probabilities would all be 1, and then this just means that you pick any of your beneficial mutations at random and you go there. So you don't actually care about the selection coefficients, right? So the random adaptive walk means that all beneficial mutants are equally likely. So all steps to beneficial mutants are equally likely. Okay, so that's all I wanted to say about these more dynamical aspects. So obviously, there's more to say about other dynamical regimes. So Daniel explained to you this regime of clonal interference. You could ask what happens if you put a population with clonal interference onto a rugged fitness landscape like this. And here I can refer again to one of the papers in the reading material. So the last one that we wrote with Ivan Sendro a couple of years ago is a study of that. And you find that, as you increase the population size, the first things become more deterministic. And then at very large population size, they become again less deterministic and so on. So there's all kinds of interesting stuff going on. And, I'm sure this will keep us and you busy for many years to come. Okay, so thank you.